Chapter 6, Reproduction at a Cellular Level. All living things must reproduce at least once, unless of course they die prior to reaching that point in their life cycle. We're going to focus on the things that live long enough in order to do that. In A, you can see a sea urchin beginning its life. It was a single cell, and now it's dividing into 2 cells. In B, it has reached its 16-cell stage as seen in the scanning electron microscope image. And then over in C, you have a mature sea urchin. There's a lot of genetic controls that goes into each one of these steps and what cell is going to become what part of the body, and we'll get to that in some later chapters. What we're going to focus on in this video is how we do step A. How do you turn one cell into 2 cells? We'll focus separately on how you do this in someone who's an eukaryote and someone who is a prokaryote. So let's focus on the eukaryotes first because they're a little bit more complicated, so it's going to make the prokaryotes a breeze. So in a eukaryotic organism, the main difference, of course, is that they have that nucleus that contains some genetic material. Specifically, in human beings, there are 23 pairs of chromosomes. In females, you can say 23 pairs of homologous chromosomes because their 23rd pair are their sex chromosomes, and women have 2 X's. Biological males have an X and a Y. You can see all the chromosomes lined up here in an arrangement called a karyotype. Chromosomes 1 through 22 are somatic chromosomes, or body cell chromosomes, and they deal with everything else that makes you you, and we organize them in grouped pairs from 1 to 22, judging by how long they are. We can use these karyograms for a variety of things, and again, we'll hit that later, but it's this genetic material that makes this process take a little longer than it does in prokaryotes, and it makes it, we have to add a few more checkpoints because we want to make sure we get this part right. Because if we give the wrong DNA copy to the next cell, then there is no point in spending all that energy in dividing because the cell's not right and it might actually make you sick. So how do we kind of gear up for that division, and what do we have to do to get there? We can look at that in the cell cycle. This is the cell cycle. It signifies the whole lifespan of one entire cell. Most of the life cycle is spent in interphase, about 90% give or take. Interphase is split into 3 sections of time for ease of study. In G1 or gap 1, we're looking at a lot of cell growth and collection of materials for protein synthesis and the eventual DNA replication, which takes place in step 2, DNA synthesis, or S phase for synthesis phase. So in this phase, we're going to replicate our DNA and then replicate the centrosomes. If you remember all the way back to chapter 3, the centrosomes are the organelles that dictate the creation of the mitotic spindle fibers and the cytoskeleton that helps the cell to move all of its internal parts during this process of cellular replication that we'll get to in the mitotic phase. The final part of interphase is G2. G2 involves further growth, further protein synthesis, and more collection of energy particles and materials that we might need for this mitotic phase. The mitotic phase is split up into mitosis and cytokinesis, and at the end of it, we have 2 daughter cells that are identical to each other and their parents. So let's take a look and see what's happening in mitosis. Animal cell mitosis is divided into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. Here you can see it with light microscopy thanks to fluorescence, or the ability to make something fluoresce or glow. Mitosis is generally accompanied by cytokinesis, which you can see on the end in that TEM picture. In some of your resources, they're going to cut out that prometaphase, and they're going to push all of those features into prophase. I point this out not to confuse you, but just to let you know, when you look at other resources, if they don't show prometaphase, it's simply because that's the way that book's written. Not all scientists include it. Not everybody always agrees. So anyway, let's look at these individual stages. So the first stage is prophase. And in prophase, the chromosomes, your genetic material, is going to start to condense. Usually it's nice and unpacked and long. You have to get at those individual nucleotides to copy them and use them and make things out of them. So we don't keep it all packed up, but we start doing that in prophase. We also have some spindle fibers that start to emerge from those centrosomes that we built in the S phase. The nuclear envelope is going to start breaking itself down. It needs to get out of the way. We have to move all those chromosomes around. We can't do that if they're in a bag. The last thing that's going to happen is that the nucleolus is going to disappear. While we're dividing, we're only going to focus on dividing so we can get all of that RNA synthesis material out of our way. Next, we head into prometaphase. In prometaphase, the chromosomes continue to condense and the kinetochores appear at their centromeres. If you think about the chromosome appearance, you usually see them written or drawn rather as those X's. The middle of the X, the waist of the chromosome, is called a centromere. On either side of that waist is a kinetochore, which I kind of think of like a little protein handle. The mitotic spindle fibers are going to grab onto the kinetochore on either side of the centromere, and that's how they're going to push that little chromosome around. So that's the next step. The mitotic spindle microtubules are going to grab onto the kinetochore on either side of the centromere of each and every chromosome. Finally, the centrosomes are going to move towards opposite poles of the cell. One is going to move left and one is going to move right, and this is going to help us move all of our chromosomes correctly and become two new cells. That's when we enter metaphase. In metaphase, the mitotic spindle fiber is fully complete, all of your tracks are laid down, and your centrosomes are anchored at the opposite poles of your cells. The chromosomes are going to be pushed and pulled, kind of like the ribbon in the middle of a tug-of-war, so the chromosomes are lined up in the middle of the metaphase plate. Each sister chromatid, which is what we call each individual half of the chromosome, is attached to its spindle fiber, and we check and make sure that we've grabbed it correctly. In anaphase, special proteins called cohesin proteins that used to be binding the two halves of the chromosomes, called sister chromatids, break down. The sister chromatids can be pulled apart towards the opposite poles. Non-kinetochore spindle fibers lengthen, so the spindle fibers that weren't attached to those chromosomes are going to lengthen and push against each other and essentially push the two pieces across towards the individual poles, and that leads you to telophase. In telophase, the chromosomes arrive at their opposite poles and begin to decondense. We're going to start using that genetic material again, so we would like to unpack it. The nuclear envelope starts to surround each set of chromosome. Each set of chromosomes. It's very important that we protect that material. And we're going to start to break down all those mitotic spindle fibers. We don't need them. They're kind of in the way. And we can reuse the material that we built them with and turn them into something else. The last stage of our long process is cytokinesis, which is not technically part of mitosis, involves the physical splitting of the two cells. In animal cells, a cleavage furrow, we'll study the specifics of that in just a moment, separates the daughter cells. In plant cells, a new cell wall is simply built between the two new cells. So here's that process with that cleavage furrow. So a cleavage furrow forms at the former metaphase plate. A little ring of fibers called actin fibers has the ability to contract or tighten, and it's going to slowly tighten, bringing the edges of the plasma membrane together. Eventually, it's going to get so tight it will pinch off. The process is far easier in plants. In part B, you can see all of the little Golgi vesicles that come together at the former metaphase plate of the plant cell. They're going to fuse with one another and form a cell plate, which is essentially a baby cell wall. The cell wall will form slowly over time and become a brand new firm cell wall, and you have two brand new baby little plant bricks. Not all cells are actively preparing to divide and enter into G1S and G2. We do put a limit on how many times our cells can divide. We don't want old cells dividing constantly because errors can happen, and that's when you end up with cells that weren't made correctly. So they can enter into G0 or the quiescent stage. For some cells, it's temporary. You enter into G0. You do whatever it is that you're supposed to be doing. You secrete hormones, hold up part of the bone, share messages, and then you enter back into G1 when it's your turn. In other cases, you remain in G0 permanently. We have some cells in our bodies that once they're formed, they don't divide again. It looks like with a lot of brain injuries, you see those areas of brain that just never reform. Certain muscular injuries, those muscles never come back. And that's what you see with cells that are in G0. We have a lot of control when it comes to our cell cycle. We're not messing around with this process, because when we make mistakes, that's when you end up with cells that are incorrect and maybe even cells that have cancer. So we have a whole bunch of checkpoints that we go through. In checkpoint for G1, we test the integrity of our DNA. So we want to make sure that our original copy of DNA is correct. It's not fractured. There's no base pair errors, anything along those lines. We'll learn more about what that looks like in a couple of chapters. But in G1, we check and make sure our original DNA is good to go. In G2, we check that our second set of DNA is also good to go, and that all of the chromosomes have formed correctly with their histone proteins that help to give them their shape. And the third checkpoint that we're going to mention is the checkpoint that's at the M stage at the that's at the M stage at metaphase. We're going to ensure that each kinetochore of every sister chromatid is connected to its own spindle fiber, and that those chromosomes can be split into their two halves. We want to ensure that that splitting is going to happen. Otherwise, you're going to have a whole chromosome go to the left, which means the cell on the left is going to have an extra copy and the cell on the right is going to be missing a copy, and that can cause major illness. So these are some of the checkpoints. The information for these checkpoints can be external signals from the body. They can be from internal checks. They can be from all sorts of regions. It's really fun to study them, but that's pretty high level bio. I hope you get there someday, but for today, we're just going to leave it at that. Let's take a quick look at what happens though when we do damage some of our DNA. So in a normal cell, if you have some damaged DNA, maybe something happened because of hypoxia, so lack of oxygen. There is something called P53. It's a signal molecule. So P53 can actually cause cell cycle arrest. So what that means is that it will stop the cell cycle as soon as it recognizes that the DNA is damaged. You do have the ability to fix some DNA damage, so your body will attempt to do that. You'll use these humongous protein complexes that are very cool to attempt to fix the DNA. If you can fix it, your body can go right on through the cell cycle and keep doing what it was doing. If you can't fix it, however, that particular cell will go through something called apoptosis, which is programmed cell death. It's a very methodical pulling apart of that cell. So you can harvest its component parts because there isn't anything wrong, and the rough ER is fine, the Golgi is fine, the lysosomes are fine. It's just the DNA. So we'll pull that cell apart on purpose and recycle everything. An issue, however, comes from when P53 is mutated. So you have DNA damage and some cell cycle abnormalities due to hypoxia, but because your P53 is missing, the cell cycle continues. And those cells grow even with their damaged DNA and can eventually become cancerous. We have found through extensive research in cancer biology that the vast majority of cancers, this genetic control known as P53 is mutated. So there's a lot of research that goes into looking at the abnormal P53 protein and trying to figure out how to correct it or maybe insert new P53 protein into sick patients so their own bodies can utilize this new P53 protein and correct any issues that they might be having. Again, we'll hit on cancerous growth when we talk more about genetics in just a few chapters. We'll end this video with binary fission, which is how prokaryotes undergo this process. And you're going to like it a whole lot more than mitosis, I promise. So in prokaryotes, remember, they have a single circular piece of DNA. They might have other tiny little pieces called plasmids, but we're going to focus on their main large piece of DNA. Replication in the circular prokaryotic chromosomes begins at one little origin of replication, and then it continues in both directions around the circle. I realize this is going to sound really funny, but think about a yogurt cup. You start to open your yogurt cup at one little point where you pull the lid, but then the lid peels back on both sides of the yogurt cup until it comes off at one point on the other side. That's how prokaryotes go about copying their DNA. It's very easy because it's just one piece. Then the cell will begin to elongate. It has circular cell and it essentially becomes this egg shape. There's a protein called the FTSE protein and it's going to migrate towards the midpoint of the cell. Your duplicated chromosomes will separate themselves and then continue to move away from each other towards those opposite poles, towards your opposite ends. That FTSE protein that migrated towards the middle of your cell is going to form a ring around the midpoint of the prokaryotic cell between your two chromosomes that you just moved over. The ring will essentially direct the migration of the outside of the cell. It's going to pinch in and form something that looks like a cleavage furrow, but it's actually not that advanced. Little plasma membrane and cell wall materials will begin to accumulate where that footsie ring has been pushing, essentially applying pressure, and you will just control a new septum or area between the two cells. Eventually, once you've built that new area between your two cells, the whole process is complete. The cell pinches into two, and you formed two new daughter cells. So it is way easier than anything else that we've been studying. Those FITS proteins that you were using will disperse. Some will go to one new cell, some will go to the other new cell, and that's it. So binary fission is just a little bit easier to study than mitosis. And with that, it's the end of the chapter. So take a look at your book, take notes in your own words, do some external review. Mitosis is a pretty neat process. We have lots of supplemental materials for you, and then go ahead and take a crack at your homework.